Good morning, Grade 8s. Welcome to the WorksheetCloud Grade 8 Natural Sciences lesson. If you have a question during the lesson, please send an email with your question to grade8@worksheetcloud.com. And finally, it's Friday. Hope you have that fun Friday feeling. So happy Friday to all of you and make it a great day. I am Mrs. Ernston, WorksheetCloud Grade 8 Natural Sciences teacher. Please remember to have your books and pens and paper and things to take notes during our online class today. Our topic for the lesson is going to be the particle model of matter, but we're actually focusing on scientific method with particular reference to learning tabulating skills. You are going to be comparing the diffusion of particles in a gas and in a liquid, and we're going to have a look and confirm our understanding that the movement of particles of one substance between the particles of another substance is called diffusion, and that particles move from an area of high concentration to an area of low concentration. So I've got a little activity here that I'd like you to engage in. So we have got a collection of pictures, okay? So I'll go through and explain each of the pictures to you so that you have a better understanding of what they mean. And then I would like you to put these pictures in order following the steps of the scientific method. Okay, so this here would be a question mark. This is someone who is explaining some graphs and tables and writing things down. Here we've got someone carrying out an investigation. Here we've got a collection of graphs and tables. Here we've got a step-by-step process. Here is a pencil that is referring to sort of a final outcome. It's the final thing that you're going to write down. Here we have three things linked together. We're going to write something down, but it's linked to a question, but it is a new idea that you have. And here is using skills of touch, taste, sight, smell, and hear. So let's pause the video and see if you are able to put these pictures in order according to the scientific method. Okay, so one here we start off with observation. Okay, so you are either going to observe something. You've either seen something or tasted something or smelled something or heard something or touched something, and that caused you to start wondering about things. So that wondering led you to being questioning and having a questioning, inquisitive mind. Once you came across a question or had developed a question, the next thing you would have done is you're going to develop a hypothesis. And then you are going to use an experiment to carry out the hypothesis. So then you need to develop your methodology. So that is a step-by-step sequence of events that you are going to follow using a collection of materials that hopefully is going to give you a set of results. So once you've got your methodology, you actually need to, let's just move this out the way, you actually need to carry out the experiment. So here we have an individual carrying out the experiments. Once you have done that, experimentation, you're going to have to present your data. So decide whether or not, is it going to be a drawing, is it going to be a table, is it going to be a graph, How are you actually presenting this data? And then you're going to have to analyze your data. So that's where you're going to have to look at your graphs and tables that you've drawn, and you're going to have to write down and link all the information together. And then the last thing you are going to do is write up a conclusion. So I'm not too sure if you want to pause the video now and write down the sequence of steps and events so that you remember it. So I'm not actually assessing the scientific method today. I was just going through each of the steps with you, and the skill that we will be focusing on today is tabulating skills. So if we do have a look at the major scientific investigative process skills that your teacher is going to want you to do, are you able to communicate or write about a topic? Can you come up with a question, or can you write a question down? Are you able to make a prediction and a hypothesis? Are you able to plan an investigation? So can you write out what materials you need and what method you are going to follow? Are you able to carry out the experiment? Are you able to collect all the necessary data? Are you able to record the data? So are you able to do a graph or a table or biological drawing or whatever is needed? And then are you able to evaluate and communicate the results? Are you able to discuss the results and analyze the results? And are you able to formulate a conclusion? So these are skills that your teacher is going to require you to learn throughout the course of the year. So we're not assessing any of the skills today. We're just going to be practicing these skills. And then you can have a look at our worksheet attached to this lesson. You will find the outline of the investigation, and the memo will be attached to that. So what we're going to be exploring today is we are going to compare the diffusion of particles in a gas to those of particles in a liquid. So the first thing that I'd like you to do is, well, the first question we're going to look at is, do particles diffuse or mix faster when they are in a liquid state or in a gaseous state? So which particles will mix more quickly, gases or liquids? And then, do particles diffuse faster with or without mixing? So you can make a few predictions here. So a prediction is, what are you expecting to happen? So are you expecting the liquids to mix more quickly than the gases? Or are you expecting it to be the other way around, that the gases are going to mix more quickly than the liquids? So you can write down what you predict is going to happen. So another thing that you can think about is, will stirring influence the speed at which gases mix? Or will stirring influence the speed at which liquids mix? Then you need to be able to write a hypothesis. So a hypothesis is, what are you going to do and what do you predict is going to happen? So generally in a hypothesis, you need to make sure that you are able to identify the independent and dependent variables as well as the control. So when we are marking a hypothesis, we check to see that you have written a statement. Okay? Remember, it is not allowed to be a question. You must include the independent and dependent variables in your hypothesis, and you must use appropriate scientific language. So again, just a reminder, I know you're going to use your prediction to help you write your hypothesis, but please remember, it mustn't be a question. So please don't start with what, when, why, because then you're going to be asking a question. So if you find you're using the what, where, when, why, and you're having to put a question mark at the end of the sentence, Then you have written a question. We need you to turn your prediction into a statement. And remember all these 3 rules here. So how do we go about identifying variables? Okay, the variables that you're going to come across in a scientific investigation are independent variables, and that is going to be your input. What do you have set up at the beginning of the experiment? And generally, these will be different variables, different criteria. Then we have your dependent variable. This is what you are recording. So what are you actually writing down? Then you have your control. That is used to test the validity of your independent variable. And then you have something known as fixed variables. These are variables that need to be able to be kept constant through your scientific investigation to ensure that your experiment is a fair test and carried out in a controlled manner. However, today's experiment is not a controlled experiment. Okay? We are not measuring any rates of mixing of liquids and gases under the same conditions. Ours is a very simple comparison of the mixing rates. So all we are doing is comparing liquids to solids. So we just want to see how long it takes each to mix under 2 different sets of conditions. However, I am challenging you at the end of the lesson to see if you are able to design and carry out your own controlled experiments. So use the tips and hints I give you along the way. So materials and apparatus. We need to check that you have everything ready. So you're going to need a glass container, a cup, or anything. It doesn't have to be glass, anything that's transparent and not really opaque. You need to be able to see through it clearly. Then you need some type of food coloring or dye. Now, in these cases, you are able just to gently pour the liquid out the bottle, but if not, a dropper may help. But if you don't have a dropper, you'll still be able to do the experiment. You just pour them very, very carefully. Then you need some type of food coloring or ink or concentrated colored drink or something along those lines, something that's a different color to tap water that you're going to be using. Then, while you're gathering those things, you can also see if you have any vanilla essence in the cupboard at home. It looks like this. Please read carefully because the bottles between the food coloring and essence can be similar. So just check that there the bottle says food coloring and here it must say vanilla essence. If you don't have any of this, see if possibly around the house you've got room spray, or maybe you've got some spray deodorant or perfume. So just ask mom before you rush off and use her expensive perfume. Or otherwise you may have some essential oils around the house or a scented candle or anything like that. So something in essence that can be sprayed. If you are dealing with vanilla essence and a liquid type of smell substance, then you are going to need a shallow dish or a saucer so that you can pour this vanilla essence into the dish. What is also going to be really helpful is whoever is with you in the house at the moment, because you definitely do need some people to help you with this experiment. So if your siblings can help you or if your mom and dad wants to get involved, that will be great. You can also try and do particularly the first parts of the experiment on your own. So press pause while you go and collect everything you need for this experiment and gather up your family members so that they can help you out. Okay, so welcome back. Hopefully you've got everything ready. Now, let's have a look at what you need to do. So the first thing we are doing here is how fast do liquids mix? So you need to fill your clear container. So I don't know whether you've got a glass or a jug, but your clear container with tap water and place it where everyone can see it. Then you're going to use your dropper and you're going to place 1 or 2 drops of your food coloring or your dye or your concentrated juice mixture or a colored juice mixture into the water. Please do not stir. So just take the colored liquid and put a few drops into the water. Then record the time at which the food coloring was added to the water. Then look carefully at the two liquids. So look at the water and the food coloring and observe very carefully how they are mixing and write your observations down below. Allow the liquids to mix, but please don't stir. So you can record the time when the liquids are fully mixed. So in other words, when the color is uniformly spread out throughout the water. So what you can do is you can press pause in the video now and you can go and carry out this experiment, record your observations, and then come back and watch the rest of the video. The second part of the experiment is how fast do gases mix? Now, this is the part where it would be really, really helpful to have your family members around you or anyone who is in the house with you. It would be great to have them help you. So this experiment should be performed in a room with the windows closed and preferably as small a room as possible. Pour some vanilla essence into the saucer. So if you don't have vanilla essence, but you've got some essential oils, you can pour that in. Or if you've got a scented candle, light the scented candle. Or if you've got some room spray, spray the room spray. But I'll need to explain that to you as we move through the experiment. So let's just read through the whole experiment first and see how you can adapt this if you don't have vanilla essence. So record the time when the vanilla essence is poured out. So in this case, if you're going to be spraying room spray or deodorant or perfume, record the time as you spray it. Then, hopefully you have some family members to help you with this experiment. So what I would do is spread your family members throughout the room. So make sure some are close to the saucer or close to where you are spraying, and make sure other family members are a little bit further apart so that your family members are dotted around the room. And then what you need them to do is the minute they smell the smell, you need them to put their hands up, or you need them to shout out that, yay, I can smell the smell. So record the time when the first person puts his or her hand to indicate that they can smell the vanilla essence. I know what is also a good idea to do at this stage is you can actually blindfold your family members so they will not be able to see you pouring the stuff or mixing it, or what you get them to do is you get them to turn their backs on you so they can't actually see what it is that you are spraying. So give the experiment some thought. Then record the time when the first person can put their hand up, that they can smell the substance. Then record the time when half the people in the room can smell the substance, or record the next group of people that are able to smell the substance, indicating also where in the room. Then step 7, record the time when the people furthest away can smell the vanilla essence. Now, if there is enough time, you can repeat the steps 1 through to 5. But what you can do now, so obviously let a little bit of time go back. So maybe you can repeat this later on today, or you can repeat it first thing tomorrow morning again. So do the experiments in exactly the same way, but this time you must run around the room and wave your arms around the room. Because then what you're going to have a look at then is what is the difference between not mixing the gases and mixing the gases, and see if it is possible for people in the room to smell the vanilla essence. So now we need to have a look at writing your results down. Now we're going to get 2 different sets of results. One is known as qualitative results, and another type of result you can get is quantitative. So qualitative results are generally, we use descriptive words. So you can tabulate qualitative results. So this is generally when you see something, or when you can touch something, or when you can smell something, or when you can taste something, or when you can see something. So that's when you, for example, you're trying to see a color change, or you touching something to feel whether it's hot or cold, or whether you can smell a smell or not, or could you taste whether something was sour or sweet or bitter, or is what you heard, is it fizzing? Is it a loud sound? Is it a soft sound? So generally you're going to use descriptive words here, and often qualitative results we use our senses. So yes, you can tabulate these. Now if we have a look at quantitative results, quantitative refers to quantity, and quantity is a number. So you will be looking at recording a mass, or a length, or a time, or a rate, or a distance, any type of number. Now, obviously, a quantitative result you can tabulate, and a quantitative result you can you can chart it, or you can do a graph. Now, sometimes, like in today's experiment, where we were smelling something, we are able to link a qualitative result with, yes, I can smell it, to time. For example, how long did it take you to smell it? Or we could include it with distance. How far away were you from the smell? Okay, so there are ways that we can take qualitative data and still analyze it. So what I would like you to do is I would like you to record any results and observations from part 1 and part 2. So write down any observations you had. And then I want you to see if you are able to tabulate your results. And I want to see maybe you are able to develop some type of charts or some type of graphs. So for today's activity, my bigger focus is actually on tabulating skills. So I would like you to see what information you are able to give me. So if we have a look at the experiment, we started out with 2 glasses. There we added our food coloring. So was it possible to predict the direction in which the color would swirl? Okay, so in this picture, we can see a yellow liquid being added to a colorless one. So notice how the yellow liquid swirls and spreads out as the yellow particles mix with the colorless particles. So of course we cannot actually see the different colored particles and the water particles, but we can make a macroscopic observation of the process. So eventually they spread out more. So what will the mixture look like when the colored particles are uniformly spread out amongst the water molecules? So I am hoping that that's eventually what your part 1 experiment looked like. So what actually made these 2 liquids mix? So firstly, what did you observe in the container immediately after the liquids were added? I want to see if you can try and put that into words. What did you observe? So using your eyes, what did you see immediately after the liquids were added? Then here we can bring in some quantitative data. How long did it take for the liquids to be fully mixed until the color was uniformly spread out throughout the water? So hopefully you were able to record the experiment from just before you added the food coloring until the end of the experiment. Then if we had a look at part 2, when the air was not mixed during the experiment. So how long did it take until the first people smelled the vanilla essence molecules? So how long did it take for those people closest to the plate of vanilla essence to smell it? And how long did it take until the last people, which means the people furthest away from the plate of vanilla essence, how long did it take for them to smell the vanilla essence? And then if you get to this experiment tomorrow, when the air was mixed during the experiment, how long did it take until the first people smelled the vanilla essence? And how long did it take until the last people smelled the vanilla essence? So what you can do now is you can design a table that compares part 2, so when we're speaking about the air, when we didn't mix the air, and when we did mix the air. So remember, how are you going to mix the air? Well, you're going to have to run around the room waving your arms. So what I'd like you to do is draw up a table with your results for the vanilla essence experiment. So just a few tips and guidelines that you need to remember when drawing up a table. 1, remember to give your table a heading. And you can't just say heading 1, a table. Okay? It must describe both variables. So it must describe your independent and your dependent variable, which means it must describe the fact that the air was not mixed and the air was mixed. And then your table must also include the first people to smell the vanilla essence and the last people to smell the vanilla essence. It's also very important to use grid lines, which means please do not draw the lines of your table freehand. Please use a ruler. Now, it is also a good idea to do that in pencil. So if you make a mistake, you are able to erase it. Then as you're putting your grid lines in, remember your table needs rows and columns. So plan what you want your table to look like before you actually do it neatly. You can choose your own appropriate column and row headings. Then just remember, it's always helpful to put the independent variable in the first column. Remember to ensure that the blocks in the table must all be filled in. Don't leave anything blank. Then you are not allowed units in the body of your table. The units must be in your row or column headings. And then please remember that the data is filled in. Don't leave your table blank. And then make sure it's nice and it's neat and it's done with a ruler and it's not all smudgy, and it's work that you are proud of. Then we're going to go where I need you now to analyze and evaluate what you have done. So you're going to write this up as a paragraph. So I need you to make reference to your graphs and tables, and I need you to write descriptions of what is going on in your graphs and tables. So what did you notice about the air and the people that were closest to the vanilla essence and the people that were further away from the vanilla essence, and whether there was a difference between mixing the air or not. Comment on, did anything go wrong in the experiments? And then can you think of anything that you could improve? So if you had to repeat this experiment or do it over again, how would you do things differently? Then I need you to write down your conclusions. So what are your conclusions? So what are your answers to your investigative question right at the beginning? Was your hypothesis proven correct or incorrect? And then state your main findings again. So what was the most important thing that you learned or that you could conclude or that you were able to analyze from your results? Then just an explanation of the content that was involved in today's lesson. Diffusion is the movement of particles from a region of high concentration to a region of low concentration. So if we want to elaborate on today's activity, what you can do now is how could you redesign this experiment to measure the rates of mixing of liquids with and without stirring? Or how can you redesign this experiment to measure the rates of mixing of gases with and without? So here I'll need you to write your investigative question, a hypothesis, a prediction, an aim, and a method. So you will find the activity with the memo for today's lesson with our WorksheetCloud worksheets that will be posted with today's lesson. So go and have a look, mark your classwork from the prep today, and see how well you did. And I look forward to seeing you next lesson for our final lesson on particle model of matter, where we are looking at phase changes. So just to recap, what did you learn today? You learned about the scientific method and the process skills. We focused particularly on tabulating skills. You were able to compare diffusion of particles in a gas and in a liquid. We had a look at the movements of particles of one substance between the particles of another substance. So we looked at the movement of food coloring or dye through water in a liquid. And then in a gaseous state, we had a look at essential oils or vanilla essence and how it moves through the gas particles in the air. So basically, particles will move from an area of high concentration to an area of low concentration. And then just to end off, I think that it will be really, really valuable, let's move out the way, for you to remember the scientific process skills. It's important that you also remember the order. So you can pause the screen now to jot the images down, but it might be easier if we just add the steps in for you. Okay, so there we can add the steps in for you. So you can start off and you can write them down in order. So 1 is observation, 2 is question, 3 you develop a hypothesis, 4 you write down your methodology with materials and apparatus, 5 you carry out the experiment, 6 you present your data, your graphs, your tables, 7 you analyze your data and you write a discussion, and 8 you do a conclusion. And then just to remember what criteria are assessed when we have a look at tabulating skills. Okay, so you can pause the screen now if you need to, and you can just copy these skills down. So grade 8s, hope you have a fantastic, fabulous, fun-filled Friday. Go out, make it great, and I hope you have a magical weekend. So thanks for watching, grade 8s. I enjoyed my lesson with you today. Hope your family members enjoyed helping you along with your scientific investigation. And most of all, I hope that you had fun. This lesson was brought to you by WorksheetCloud.